Hi friends, welcome to my video. In this video, we are going to see about bacterial transformation. Transformation is the direct uptake, incorporation and expression of foreign DNA or genetic material from the environment. Bacterial transformation is a major mechanism of horizontal gene transfer. Let's see, what is horizontal gene transfer? Horizontal gene transfer is the non-sexual movement of genetic material between unicellular organisms or between unicellular and multicellular organisms or between multicellular organisms. Incoming genes can replace or introduce new genes into a genome. That is, horizontal gene transfer is a process in which an organism transfers genetic material to another organism that is not its offspring. Horizontal gene transfer, also known as lateral gene transfer. Horizontal gene transfer is an important factor in the evolution of many organisms. Bacteria have acquired a variety of important traits, including antibiotic resistance, pathogenesis, and metabolic pathways via horizontal gene transfer. Next, we are going to see what is competence. Under extreme environmental conditions or under specific growth conditions, in some bacteria, the coordinated expression of several genes allows bacteria to import foreign DNA. This specific situation referred to as competence. Let's see the definition of competent bacteria. The bacteria that can take up the foreign DNA from the environment by transformation are known as competent bacteria. There are two forms of competence, natural and artificial. Natural competence. As I stated earlier, natural competence refers to a specific physiological state in which bacteria are able to take up free DNA from their environment. Artificial competence. Artificial competence can be induced in laboratory that involve making the cell passively permeable to DNA by exposing it to conditions that do not normally occur in nature. For example, by chemicals, by applying a strong electric field, by using a heat shock. We should note one important point here, that is, not all bacteria are capable of taking up foreign DNA from their environment. And several bacteria can be artificially treated in the laboratory to increase their transformability. We learned what is competence. Next, we are going to see some more details about transformation. Transformation has been studied mainly in pneumococci, bacillus species, and Haemophilus influenza. Pieces of DNA involved in transformation may carry about 10 to 50 genes. The process of gene transfer by transformation does not require a living donor cell, but only requires the presence of free DNA in the environment. Once the DNA enters the cytoplasm, it may be degraded by nucleases or may integrate into the chromosome. Transformation works best with DNA from closely related species. The naturally competent bacteria carry sets of genes that provide the protein machinery to bring DNA across the cell membrane. You should note one important point here, that is, only single-stranded DNA may pass through. One strand is therefore degraded by nucleases. The translocated single-stranded DNA may then be integrated into the bacterial chromosome. One more point to be noted here is that due to the differences in structure of the cell envelope between gram-positive and gram-negative bacteria, there are some differences in the mechanisms of DNA uptake in these cells. DNA as a nutrient source. DNA is an excellent source of the deoxyribonucleotides needed for replication of the cell's own genome. Environmental DNA is ubiquitous in most bacterial environments, especially biofilms. Competent cells take up preformed bases and nucleotides where possible and also use extracellular DNA as a nutrient source. Most competent bacteria that have been tested can take up DNA from any source. Let's see the sum up of bacterial transformation and competence. DNA has both biochemical and informational properties. The effects of DNA uptake depend on the nutritional needs of the cell, the presence of DNA damage, the ability of incoming DNA to recombine with chromosomal DNA, that is, high similarity with the chromosomes also allow extrachromosomal DNA to change the cell's genotype by homologous recombination, a process called natural transformation. Otherwise, it is degraded and its nucleotide subunits are recycled. Thus, competence provides cells with a potent mechanism of horizontal gene transfer and nutrients. Up to this, we learned what is bacterial transformation, horizontal gene transfer, and competence. Next, we are going to see about Griffith Experiment. Transformation in bacteria was first demonstrated in 1928 by an English medical officer, Frederick Griffith. Griffith was researching about the distribution of pneumococcal types. Griffith conducted a series of experiments using Streptococcus pneumoniae and mice. In his experiment, which is popularly known as Griffith Experiment, Griffith used two related strains of bacteria known as R and S strain. Let's see what is R strain. When grown in a petri dish, the R strain formed colonies that had a rough appearance. R stands for rough. The R strains were non-virulent. That means they did not cause disease when injected into a mouse. Let's see what is S strain. When grown in a petri dish, the S strain formed colonies that had a smooth appearance. S stands for smooth. The smooth appearance was due to the presence of a polysaccharide capsule. This capsule protected the S strain from the mouse immune system, making them virulent. That means when injected mice with this S strain, the mice developed pneumonia and died. Friends, up to this we learnt what is R strain and what is S strain. Next, we are going to see the outline of Griffith experiment. Griffith took two groups of mice. For the first group of the mice, he injected R strain, and for the second group of the mice, he injected S strain. Mice injected with R strain lived, and mice injected with S strain died. We know the reason, because R is non-virulent and S is virulent. In the next part of the experiment, the mice were injected with EAT-killed S strain. The mice all lived. Here also we know the reason, because he used EAT-killed killed virulent strain. In the third part of the experiment, the mice were injected with a mixture of killed S strain and live R strain. From the previous experiment, we learned that the strains used alone were unable to cause disease. But surprisingly, the mice all died. Blood sampling showed that the blood of the dead mice contained both live S strain and R strain. Because the original virulent strain was inactivated, Griffith concluded that the non-virulent R strain had transformed into the virulent S strain. Griffith postulated that information, which he called transforming principle, could somehow be transferred between different strains of bacteria and converted the R strain into S strain. We should remember one important point here, that this was long before the discovery of DNA as a genetic material. Griffith was not able to identify the chemical nature of this transforming principle. The nature of the transforming principle was identified as DNA by Avery, McLeod and McCarthy in 1944. Thus, Griffith experiment led to the discovery of DNA as the genetic material. While explaining the Griffith experiment, I told you Griffith used killed S strain and live R strain. So you may ask me how DNA from killed S strain convert R strain into S strain. The answer is that DNA molecules can renature after treatment. Renaturation is the reconstruction of a nucleic acid to their original form, especially after denaturation. I'm concluding this video here and see you soon in my next video. Thank you, friends.